Get started? I guess so. All right. Welcome back, everybody. We are joined here by the Cancer Insight team. We're continuing our regulatory and clinical trial today. This session is going to be really a lot more focused on trial design, implementation, and key considerations when you're thinking about setting up your early clinical trials. Really excited to be joined by George Peoples, Dr. George Peoples, who helped found and is the CEO of Cancer Insight. Dan Hargrove, who is president of Cancer Insight and huge supporter of the program, and Lori, who's COO of Cancer Insight. So we have full leadership here, and they have been growing like crazy over the last number of years. Worked with a tremendous number of the startups and smaller biotechs, mid-sized biotechs all across the state and all across the country. So with that, I'm gonna let you all sort of take it from here, and really excited to have you back with us.
Dan, did you want to give any quick background? I will jump right into the content, but would like to give you just 2 minutes on Cancer Insight.
Sure.
Dan, did you want to?
Yeah, let me jump in real quick. So I've met most of you all on the call, and it's been my pleasure to help you. And so as we've discussed in the past, and for those who have not met us, Cancer Insight is a specialized clinical contract research organization. And we got our start really with taking George's academic program out of the military. So George is a surgical oncologist by training, but has been doing drug development, especially cancer immunotherapy, therapeutic development for about 30 years. And so when we spun out his academic program out of the military, George had this drug development team. We maintained that focus of translating novel oncology products from the bench through the FDA, and designing those complicated first human clinical trials. And fast forward now, we're doing phase 3 studies, and so we're a good operation. We're up to staff of over 60, have several oncologists on staff, full service. And so it's been our pleasure to work with the ACT program. We are a huge supporter of the ACT program. We appreciate and understand the struggles that you all have, but importantly, this is really important. We admire what you're trying to do to translate your product and bring it to the marketplace. So with that said, I'm going to turn it over to George and Lori for the remainder of the program. I'm unfortunately going to have to drop off at 12:30 because I have calls. George, over to you.
Thanks, Dan. So Lori and I are going to kind of tag team this. So just kind of set the stage. I'm going to give you more of the theoretical, considerations of, how we get to, the specific clinical trial designs. And then Lori's going to do it much more practically, the steps you have to take to implement some of these trials. So just to kind of supplement what Dan said, I come at this very clinically, practicing surgical oncologist for 25 years, ran a number of multidisciplinary cancer teams and programs. And so once again, I'm always looking at this from the perspective of the patient, the cancer types, the current treatment landscapes, where we stand right now with the NCCN guidelines. And so there are a number of companies that have approached us really to help kind of give them a bit of a game plan, if you will, map out the clinical development strategy, and then go to trial design, protocol development, and of course we can run studies as a full service CRO. But we're here to help. There's other groups, obviously, that you can approach that have similar offerings, and so we're not the only show in town. But this is kind of our sweet spot, if you will, because we've developed a number of products of our own. So we've been sitting where you are as the developer, and then of course we've helped several dozen companies with their plans. So with that as a little bit of background, well, actually, before I kind of go to the presentation and get into some of this, any questions right off the bat for Dan or Lori or myself about Cancer Insight, or any questions you have just in general, and then we'll jump into the content. All right. So let me, here we go. I'm just going to share this real quick. All right. Can everybody see?
Yes, we can
More
see your screen,
slides?
George.
Okay, perfect. Let me. All right.
Or just, we are looking at the PowerPoint view. I don't know if you wanted to put it into presenter view or not.
Yeah, that's what I was going to do. Make that a little bit easier. Does that look better?
It's great.
Okay, perfect. Now, guys, this is meant to be interactive. And so I'm going to start going through this, but if you have questions, please, feel free to jump in and give me a yell, and we can answer things as we go along. Once again, we're going to start with more the theoretical on considerations in the clinical trial design, and then ultimately, like I said, Lori will kind of take over and talk about more of some of the practical elements. So when we talk about, clinical trial design, and I'm just going to assume right now that you guys know the general consent or the general, organization of studies. Of course, phase 1 trials are meant to be oftentimes first in human, but then also are meant to be for safety, primarily dose escalation type studies. Phase 2, we begin to think about efficacy. And then of course, phase 3, we're trying to prove that against the current standard. Those are kind of just the basic phases of studies. Now, of course, there's a lot of, additional types of categorizing trials from 1B trials to 2A trials to 2Bs, stage zeros. And so there's more there to it. But what I'm really going to be focusing on here is your initial clinical design, because I'm assuming the majority of you are early in your process. You have a technology, you're starting to think about what your path is going to be and what that first clinical trial is going to look like, excuse me. So that's the approach that I want to take here. So the elements that we'll talk about over the next half hour or so, the basic elements of the clinical trial design, the interplay between those elements, some of the innovations that we're starting to see in clinical trial design. It's not your parents' clinical trials anymore. We're really trying to get more sophisticated, more efficient in the way we do clinical research. There are special considerations, particularly for certain classes of drugs like IO. And I emphasize that not only because this is kind of our specialty area, but also, as many of you are fully aware, your products are going to have to fit within the current landscape. And the current landscape is being dominated right now by new IO products. How are you going to interact with them? How are you going to combine with them? How are you going to compete with them? And so there are some special considerations around trial design that incorporates some of these IO concepts. And then I do want to leave a little bit of time for your questions and any discussion that we have. We can talk a little bit about the theoretical in between, or we can wait until Lori's finished with her implementation part and we can have a more general discussion. But once again, please feel free to interrupt me as we go along here to talk about any specific questions you might have as we go. So the trial design process starts with your technology, okay? And once we have some more understanding of that, and we're going to go through these individually, but that's what we have to understand first. What's your technology? What is it? What does it do? How are we going to try to deploy that? Then that's going to help inform what the overall trial design is going to look like. But then we get into the specifics, like what is the specific target patient population that's going to be utilized for that study? And then, of course, ultimately, what are the endpoints of that study? But when you really get down to it and you look at all clinical trials that are being run, you can boil it down to these elements. But of course, there's a lot of interplay between these, because if you change, technology, well, it may necessitate changing the trial design or the target patient population. If you change the target patient population, it may end up changing your endpoints. You get the gist. So technology, what do we know about it? Okay, you are the expert, it's your technology. And so if you were to come to us, one of the first conversations we're going to have is around what is it? What does it do? How does it work? Okay, what's the mechanism of action? Is this going to be ultimately delivered as a systemic drug, a local drug? Is it IV, or is it, a vaccine that is an intradermal injection? Based on the mechanism of action, are there expected toxicities? We work with a company that has a risk for cardiotoxicity. So how are you going to give this drug? How are you going to dose it? Ultimately, when you design your trial, you have to take into account how you're going to monitor for those specific toxicities. And then what's the expected outcome? I mean, are you looking to prolong, progression-free survival? Are you looking for, objective responses where you're trying to shrink tumors? Is it a cytotoxic agent? Is it a cytostatic agent? Okay, so all of those elements have to be understood because they all play ultimately into your design. But once we've answered those questions, we can start really putting pencil to paper and start coming up with the concept. And just really quick, brief digression here. When we do this kind of work, and once again, happy to engage with any of you to have these kinds of discussions. But once we go through this, the process we do is understand these elements, put together literally a one-page protocol concept sheet or trial concept sheet that says, here's the target patient population, here's the drug, here's how it's going to be used, and here's what our endpoints are going to be. And then from that one pager, then we can start fleshing it out into a synopsis, and then ultimately from a 5 to 7 page synopsis into a protocol that is ready to be submitted to an IRB. So it is an iterative, additive building process that we go through, but it all kind of starts with this ultimate discussion around the technology. So once again, talk about the technology, then we get to the design. So we want to match the design of the trial to this mechanism of action and, quite frankly, the stage of development. If you tell me this is a product that we've already got some basic safety information and we're ready to start looking at an efficacy trial, do we do a 1B, do we do 2A? How will that look? Is it single-armed? So those elements are going to be different based on the stage of development. Of course, the assumption here is the majority of you have a technology that hasn't been tested yet, and so it's going to be a first-in-human study. Well, what does that look like? And the type of trial, obviously, as you've already mentioned, phase 1's primarily around safety, phase 2 is around efficacy. However, even in a phase 1 trial, you can begin to get some efficacy information. Hence the reason why we call some trials 1Bs, because it really is about safety and dosing, but we're going to collect some efficacy information. The target population oftentimes is dictated by the technology, i.e. we have a small molecule that blocks a pathway. That pathway is only expressed in a certain patient population, maybe even a specific cancer. Okay, well, that's all dictated to us. But more frequently, you will have the entire landscape of cancer to choose from. So, where do you go with your drug? And so we'll talk a little bit about that on the next element with the target patient population. But depending on that patient population, that's what's going to dictate your endpoints. Are you in a disease-free host versus are you in a patient population that has metastatic cancer, and therefore, your endpoints are going to be more like objective response rates or clinical benefit rates. To this target patient population, what patients, okay? What type of tumor? What stage should it be? Are there subsets within those tumors? And once again, getting back to, small molecule, specific pathway, you need to know that that pathway is important to that tumor for that patient population to have a chance of responding to your drug. So sometimes this is pretty straightforward, other times it's not, because maybe your drug's more generalized. and you could go to a number of different types of tumors. But once again, we're going to match that patient population against not only your mechanism, class of drug, but also the stage of development. Match to the trial design, because once again, is it a disease-free host or is it a metastatic patient? You know, are you looking primarily for safety in metastatic patients, or do you need to go to a patient population with a lower tumor burden to be able to generate an efficacy? And then quite frankly, sometimes it comes down to strategy or the tactical elements. And what I mean by that is, let's say you have a drug where you can go to a number of different types of tumor. So which one do you choose? Well, there's strategic reasons for choosing a tumor because it's a bigger market, it's an unmet need, there's some huge gap there that can be filled. So there is strategic reasons to go into a specific tumor type. But quite frankly, from a trial perspective and the actual implementation, enrollment, and execution of that study, there's a tactical element that has to be addressed. Are these patients available? Can I access them? Can I get my trial done in a reasonable time frame? So there's additional considerations there when you're choosing your target patient population. Now, of course, the endpoints. Yes, please.
Before you move on to endpoints, it would be interesting, sort of as a case study, a recent client we've had, you know, having to sort of go back to the drawing board because, you know, they generated all of their preclinical data using models for, you know, triple negative breast cancer. And then, you know, when it got time to get to their IND stages, and they came to us and, you know, we sort of did some initial feasibility and we're like, look, you're not going to be able to get the tumor tissue that you need. You're not going to be able to enroll because of competitive landscape. And it set them back quite a bit because now they're going back and having to do preclinical work in different animal models. So this is kind of what I like to call, if you haven't heard about it, this acronym before, it's called your TPP, your target product profile. And, you know, from the minute you have an asset, you should be thinking about these things, like thinking about keeping the end in mind. I know it's very exciting when you get something and you just want to, you know, go, but putting some of the upfront work and considerations may save you time and money in the long end to get your product to market. So think about your target product profile, who your intended target population is going to be, where they're located, how that fits into the current standard of care, and ultimately into, you know, all of these things that George is talking about. So George, I don't know if you want to comment further on that little, you know, sort of case study, but.
Yeah, and I think that's perfect because that's a nice practical example of what I mean between strategy and tactics, right? The company in question that Lori raised is more of an autologous-based company that's dependent on being able to access the patient's tumor, and hence the problem. Okay, so while, you know, triple negative breast cancer is a great strategic target, from a practical sense, it was just not feasible. So the tactical element had not really been considered. So I guess the takeaway from both Lori's comment and this point, it's never too soon to start thinking about what your clinical development plan is going to be. What is your TPP going to look like? Because it can inform a lot of your strategy and quite frankly, what has to be done to get you to your IND, through your IND, and into your first in human study. So once again, we'll open that up for discussion here in a bit. Go ahead, please.
So can DMCI Act through this program, can we get you know, advice on TPP? You know, what patient populations are available, or you know, I think Sarah sent me some information on related to earlier, but more structured information on each of the drug candidates that we have. That information will be extremely useful.
Okay, can I jump in real quick, George?
Yeah, please, sir.
Yeah. You guys have been just setting this up so wonderfully for us because we're actually going over TPP in more detail later today. That'll be the later afternoon session, thinking
Okay.
with the strategic angle, and this is part of what we're going to be working on over the next couple of months. So we're going to talk about that in more detail. I don't want to derail the conversation that's ongoing, but just to give you a little bit more perspective, part of that process is talking to people like George and Lori, and also to KOLs, as you probably gathered from the way they were sort of describing this, and developing that perspective more thoroughly through that effort as well. So it's not something we're going to have all the answers for, but we're certainly going to talk to the process later today.
Okay, thank you.
And probably also as a part of that conversation, you will realize that, and I know one of the end goals of the program is to get you to the point where you can submit like to a CPRIT application, and CPRIT requires you to submit a TPP. They want to see that because they want to know that you've given it ample thought and consideration. So once again, something we can discuss in more detail later. Endpoints, important in a lot of times is going to be obviously matched to the trial design. Phase 1 is going to have different endpoints than a phase 2. But even within that, how you're going to look at your safety and what are those safety endpoints going to look like. There's obviously usually dosing, some PK/PD work that's going to go into it to determine what your recommended phase 2 dose is going to be for your efficacy studies. Now, it also is going to be matched to your patient population. Once again, is this a metastatic population which has measurable disease, and therefore you can do an objective response rate and look for complete responses, partial responses, stable disease, et cetera? Or is this a drug that's going to work better more in the preventive setting, where it may be a cancer patient who's had definitive therapy and you're trying to prevent their recurrence or progression, and therefore that's going to dictate what that endpoint is. And then ultimately, and of course, we're talking early phase trials here. So, but to skip ahead as you're thinking, about how we go through the safety and efficacy, and we eventually get to approvable type endpoints, the gold standard still remains overall survival. However, there are a number of conditional approvals that you can receive from the FDA. For example, you can receive a conditional approval based on PFS or DFS in certain disease indications. You can get, accelerated approvals based on ORR or durable objective responses. And of course, all of these are based around RECIST criteria. Now, I would hope that the majority of you have at least had some experience or exposure or understanding of RECIST. These days in oncology and the way we measure whether or not a drug's working or not early to see tumor shrinkage is all built around this now fairly standardized system of RECIST, current version 1.1. But because we also have a lot of emphasis on IO type products, do understand that there's also a variety of variations on the RECIST concept that are meant to be specifically around immunologic type drugs and mechanisms. The one that's used most recently, or this is the most recent version of it, is iRECIST, which uses a lot of the same RECIST verbiage and categorizations, but then applies one major difference, and I'll go ahead and mention it here. It'll come up again later. But the reason why there's variations on the RECIST criteria for immunologic products is because you are inducing immune response against the tumor. As the immune system invades and fights that tumor, it is not uncommon for that tumor to actually swell before it starts regressing. And we noticed this a decade ago now when we first started doing these kinds of studies. And of course, we called that pseudoprogression, but it completely messed up, trial designs that were used to looking at tumor shrinkage. And then we would see these tumors swell, then that would count as a progression. And therefore the trial or that patient may be considered as a progressive disease failure, when it turned out they were actually responding to the drug. And so once again, I'm sure most of you have heard all of this, and therefore just understand that we have some alterations on the RECIST criteria that are specific for immune mediated type drugs. So once again, just as a bit of an overview, very superficial, but just to understand that these are the elements that we wanted to center in on when we first start designing a trial. And if you think of it, it's fairly daunting to say, well, I've got to write a protocol for running a clinical trial if you've never done that or have had a little experience in that. It's a very daunting task, but like most things in life, you break it down into bite-sized pieces, it begins to make sense. So do understand, no one ever just sits down and writes a protocol, okay? That comes from a synopsis. The synopsis comes from a concept. The concept comes from these elements. So if you start thinking about these elements, then you are on the path to designing your first clinical trial. None of this is left to chance in the way that we actually run clinical studies. This has all been very well dictated and codified into some very specific guidances from a number of international organizations, but also the FDA, whom I'm sure most of you will be working with. So as we talk about, okay, now we have a bit of an idea around what the design is, there are some special considerations that I think are worthy of mentioning early on, just where you have this as background information and you begin to think about it going forward. Now, I'm not sure what all of yours products are, but for those of you who are in the IO world, there are some specific special considerations for IO products. I just pulled up an example. The FDA has been very clear and given us guidance for the development of specific forms. And here, this is one, once again, just as an example, you can go and find, specific FDA guidance for the clinical development of, say, therapeutic vaccines, for example. And there's, similar guidances for other classes of drugs. So please understand that the FDA has a tremendous amount of information out there. There's only a fraction of it that's going to be directly related to what you're doing or your class of drugs. So don't think of it, once again, as being overwhelming. Find the information that's most pertinent for you. These accelerated safety designs. So we've been doing clinical trials forever, not us personally, we've been doing it a long time, but the field has been doing clinical trials forever. There are kind of prescribed safety designs. Everybody's heard of the traditional, 3+3, dose escalation safety designs. Believe it or not, it still works and it's still used more often than not, but there are now ways to make that process go quicker, particularly if you have a compound where you feel that I'm going to need multiple dose escalations, okay? So this isn't, just 2 or 3 escalations. This is, multiple. I need 5 to 7 of them. Well, if you have to do your initial 3 patients, look for toxicity. I, saw toxicity in 1, so now I've got to add the other 3. I've got 6 patients in this one cohort. I've got 6 more to go. I mean, it gets a little daunting, right? So if it is a drug that has, no human data, you're having to start at an incredibly low dose. You're looking at 6 or 7 dosing cohorts to get to what you think is going to be the therapeutic dose based on your preclinical models. There are now ways to do accelerated safety designs where literally you're doing single patient cohorts at the ultra low doses to get you. And as soon as you see your first, grade 2 toxicity, then you convert to a traditional 3x3 design, for example. So I just want you to understand that the FDA is trying to work with us and help us come up with more accelerated ways of doing things. This is just the paper that helped establish some of these accelerated designs that's oftentimes quoted. And I'll provide this information for you guys. Adaptive designs. Here's a term that gets thrown around very, very frequently. And I will tell you that if you ask most people, what do you mean by an adaptive design? it'll be completely different from what the next guy thinks about an adaptive design. It's a bit of a bucket term that, just gets tossed around a lot. What I would tell you is what the concept is behind an adaptive design is the idea that there is an element where the trial can be adapted based on the initial result. Now, that adaptation could come in multiple forms. It could be that you start 3 arms to a trial and you find out that 2 of them working and one's not, and the adaptation is you're dropping an arm out of the study. Another adaptation could be, we're going to start with a small number of patients, find out what our treatment effect is, and once we define the treatment effect, that will tell us how to power our study, and therefore we'll have an adaptation where we can change the size of the study to ensure adequate powering. So once again, it can come in multiple forms, but the idea is trials do not have to be written in stone the way they were in the past. You can design a trial that allows you to modify the study as you go along. Okay, and probably the one that comes up most frequently as an example is we're ready to, we want to start our phase 3, but we don't have all of our randomized phase 2 data to, de-risk that phase 3. Can we do a lead-in phase 2 randomized that then seamlessly rolls into our registrational study? This is where we hear about it most frequently. That concept is feasible, but it is sophisticated, complicated, and has a lot of moving pieces. It's not always the right answer. This is one of those where probably 9 times out of 10, when we hear that somebody wants to do adaptive design, it never comes to fruition. They end up doing a traditional phase 2 followed by phase 3. Is it doable? Absolutely. I personally think some of the adaptations that make the most sense occur before the phase 2, phase 3, concept, when you're trying to figure out how best to use your drug, the right population. Your adaptations are more around enriching your population or powering your study. And so those are the kind of elements that are of interest. And once again, there's a number of examples of how to do adaptive design, some great reviews. This is one of them that I particularly like. And it goes through a number of different ways to do adaptive studies.
And George, just want to pipe in here also is that from a statistical perspective, there is a methodology that a lot of people get excited about and come to us and say, hey, there's, Bayesian adaptive design. And this is something that we generally don't recommend for early phase studies, but people do sort of like the idea of it. And it, it's something that floats around there, but it's called adaptive by design. But statistically, it has a very different meaning than the examples that George is talking about. George, do you want to clarify that at all?
Yeah, and to that point, Jack is a statistician. So a lot of these adaptations, adaptive designs have been generated out of the statistical groups about how best to capture the number of patients you need and increasing your odds of success in your study, okay? I mean, that's the motivation behind them. But I will tell you that some of these get quite sophisticated to the point where, it's statistician speak, right? And that, I don't know about you guys, I don't want to, undercut anybody's skill set here, but I think I understand stats at times. And then I talk to a statistician, I realize I know very little about statistics. So it can get to a very sophisticated point. Another case study, we had a company come to us and they had gone to a statistician. They had this elaborate, phase 1 study, which in the end needed to show that their drug was safe, what was a recommended phase 2, and do it as quickly as you can do it with the fewest number of patients. Okay, I mean, in the end, that was the result. Their design was so complicated that none of the sites, none of the PIs could understand it. And because they couldn't understand it, they didn't want to do it. Okay, so once again, it gets back to this whole practical element, so there is a reason to try to keep things as simple as possible. Get your design, get the data you need, get you to the next step, but do it in a way that is most efficient, and oftentimes that is in its simplest form. Okay, so we can once again discuss those elements as well. I've already touched a little bit about these accelerated approval endpoints. I only throw them out there for you right now because, and once again understand most of you very early and you're not thinking about approval, but yet to Lori's point, in your TPP, you are thinking about approval. I mean, that TPP eventually translates to your label, right? And so, how am I going to get this drug approved? The good news is there's ways to get approvals early on, and there are surrogate endpoints that can be used for accelerated or conditional approvals. And these are not necessarily, mysteries either. Okay, so once again, just pulled up an example, non-small cell lung. It's changed tremendously over the past, 5 to 10 years. And, we're seeing approvals and changes in standard of practice to the point where the FDA basically said, look, here's what we need to get your drug approved. And, you would think it's just overall survival. No, these are the other things that we think are important. Breast cancer is another good example. A lot of breast cancer now has gone to neoadjuvant therapies. Well, there's a point now where if you give your drug in the neoadjuvant setting of breast cancer and you get a PAT CR, that is a surrogate for survival. And therefore that now has, been elevated to a surrogate marker for accelerated approval.
You know what? I just thought of another sort of, warning and case study, George, and thinking about, endpoints and settings to do your trial in. And I'm thinking about my former company when we started out our development program. And one thing you want to keep in mind is the rapid change in standard of care, like in the course of your development. So when you're thinking about your TPP, and you don't necessarily want to think about, what is the standard of, practice today, but what is it going to look like, 5 years from now, once I have some meaningful data, because I work for a company, we put a lot of effort into a specific patient population, specifically patients who were naive to checkpoint inhibitors. And then all of a sudden we get, the checkpoint inhibitor approvals. And now I basically have a drug intended for use in a population that is no longer applicable or available in the United States because of the change in standard of care. So that's just another sort of tip and lessons learned that we can share for you to think about moving forward, is not just the standard of care today, but what's in the pipeline over the next, 3 to 10 years when you expect to get your approval, that, you want to make sure that you're still relevant at that time.
And I think that is, it is a great point, and it is a difficult one. The good news is there's a lot of new drugs available, and the way we treat oncology patients today doesn't look anything like the way we treated them 10 years ago, just because of the rapid advances. The converse of that is, if you're on the drug development side, it's tough. I mean, you are constantly looking forward, and it does require you to be nimble as far as how you're going to develop your drug. And you cannot get locked into a specific indication, a specific stage of patient, because you can set yourself up for failure very quickly if the standard of practice changes in that specific indication. So that's another one of those reasons why groups like ours exist, because we do keep tabs on the standard of practice, the current NCCN guidelines. We keep a very close eye on what's coming down the pike, and therefore what changes are likely to occur. Now, nobody has the magic crystal ball, but nevertheless, you can be well informed. And you can also think, and this becomes that strategy tactical type of discussion around where is the least risk for me in developing my drug? What's the area that's least likely to change? Versus, where are the real risk areas where it's highly likely that we're going to see some changes in standard practice. And of course, those are the areas you're going to want to avoid initially. Just two other quick points here. You may find that the drug that you're working on is not going to be a good standalone drug. Maybe it's a cytostatic agent, or it's a drug that works synergistically with another class, perhaps one that's approved. And so if you are working with a drug that ultimately is going to need to see combination, then there are very specific ways we design trials. Of course, the FDA still requires some monotherapy safety information, but that can be done as a lead-in where you dose your drug as a monotherapy, collect that information, and then segue right into a combination that is going to be necessary for the drug to work. Okay, and so there's ways to implement those strategies. And of course, again, not to over belabor the IO conversation, but these days, the world of cancer largely is divided into those indications where IOs are approved and those where they want to be approved. Okay, so and if you have a drug that is going to be in combination with checkpoint, do you go into an indication where there already is an IO approval and your strategy is we can make that better? But the IO drug's already there, standard practice, it's paid for by third-party payer, and all we have to do is add our drug on top. That's a whole strategy in and of itself, as compared to we're going to go where checkpoints want to go but haven't been successful yet because our combination is going to make checkpoints work or our drug work. And so now you're developing two unapproved therapies in an indication. And so that too falls under very special, guidance from the FDA. So if you have two unapproved products, how did you develop those together in order to come up with a approvable combination? And this is another paper which I found particularly helpful. It's a little dated now, but nevertheless, the points in it are very well taken, written by Jed Walchuk, who is one of the leaders of the whole checkpoint inhibitor world, PD-1s in particular out of Memorial Sloan Kettering, and literally laid out how do you do this if you're going to do combinations with immunotherapy products, and in particular checkpoint inhibitors. And so, I guess the point here is I've just pulled up examples as we've gone along, mainly to just tell you that there's no mysteries per se out there. There's information there and available, either guidances from the FDA or from the literature about how this goes. This is a very old example, but I use it to make a point. 2017, if you actually remember, checkpoint inhibitors, the PD-1s, pembrolizumab, nivolumab, were first approved in 2014. So the first approved PD-1 inhibitors, 2014. 3 years later, 2017, at SITC, 164 targeted agents towards PD-1, PD-L1. I mean, this is how quickly the field goes, but it is also a field of herd mentality. And so I was having a conversation earlier today, and God gave an example that I just loved. He's like, when you go out on a fishing boat and you're out there and all of a sudden you look over and there's just a cluster of fishing boats, it's not because that's where all the fish are. It's just everybody sees that's where all the fishing boats are, so they assume that that's where all the fish are. So there is this herd mentality that occurs once you get a successful product. And so you just need to be aware of that. I mean, do you want to be the person who has the 165th PD-1 directed therapy?
And I just want to tack on to that, because you're really showcasing how competitive it is. And when you're in, emerging biotech, it's super important that you're able to enroll your trial. And in order to do that, when you're thinking about your trial design, you want it to be patient friendly and you want it to be site friendly because there is so much competition. And to be patient friendly, you need to think about things like your visit schedule. Are you asking patients to come in multiple times a week for, for their therapy and treatments? Or is this something where they can come in, every other week? How many biopsies are you requiring during the course of your protocol and your treatment period? How many blood samples? Anything you can do to make your protocol more patient friendly, the easier time you're going to have accruing. And same thing from the site staff perspective. George already mentioned this overly complicated, randomization algorithm for the protocol design that the sites didn't like, but also, think about the nurses, like, and how many things you want to put into your protocol, how many procedures, how many, I see a lot of companies, they try to put everything in the kitchen sink into, one protocol instead of doing it over maybe 2 or 3 that they should. So they get this kitchen sink type mentality, and the sites just look at it and they're like, yeah, that's too much. And we'd rather, funnel our patients into the studies that are easier for us to conduct. So just a little, lesson learned there as well on trial design.
And I appreciate that, Lori. And as I kind of hopefully at the beginning of this, I'm giving you a lot of theory. Lori's giving you a lot of practical, and we'll continue that theme. But each one of these elements, they do have a direct correlation to what is the likelihood of you being able to conduct this trial successfully and get the data you need to move to the next phase, okay? And orphan is one that comes up frequently. A lot of times companies will build their entire strategy around orphan, once again, from a strategic and theoretical standpoint of, well, it's an orphan indication, big pharma is not that interested in it. It allows me to be able to play with the big boys, but in a small pond here, and I'm going to go after an orphan indication. I'm not saying that's not valid, because it is, and we have some of our sponsor companies that we work with that do have orphan indication, and these patient populations are deserving of research attention. Like most things, though, it is a double-edged sword. The reason why it's an orphan indication is because these patients are few and far between. We did have a company come to us one time and say, we're going to go after this indication. There's 500 patients in the United States per year with this disease. Well, good luck running a clinical trial on that indication and trying to get access to those 500 patients, okay? So just be aware that there is no magic bullets here. There is no easy answer. Each of these strategies will have its own set of issues. It's just finding the strategy that works best for you with the fewest number of issues so that you can be successful in getting your trial done. So once again, just a quick summation. We hit a few of the elements, how those things interplayed, a few special considerations on trial designs. Do understand there's a ton of information out there. You can find this on your own. We're happy to help guide you as well on this, but ton of things that are available to you to help guide the process. But also, as we mentioned earlier, having some of these discussions early on so you begin to get a feel for the playing ground that you're going to be on, is important. Thinking about your TPP, as Lori's already introduced here, we'll go into some more of that. Sounds like you guys will as well later today. But right now I'm going to pause, and if there are any questions, concerns, topics that I haven't touched on, I'm happy to do that now. And then I'll turn it over to Lori for some more practical elements.
Russell, you had a question?
Yeah, mine's a practical one. Do you want me to give it to you now or?
Yeah.
Lori's your person, so.
Go ahead.
Okay. Okay, so in regards to our IV, is there kind of a barrier, like as far as dosing is concerned, as far as frequency, like the regimen? Like, for example, if I had to go with a daily, is that like a major problem or major barrier to a phase 1?
Well, it depends. For a phase 1, you can potentially conduct the study, but you have to think about, is that going to be viable in the market? I mean, unless you're showing 100% complete responses, it would be very difficult to convince patients to come into a clinic on a daily basis. So if it's something that you're doing in the interim to get into patients while you work on an alternate dosing schedule, then that would be okay. But I would never recommend going into phase 2 with an intense dosing schedule like that.
Yeah, that answers my question. Thank you.
Did she just kill your program?
No, she validated it. Thank you.
Okay, good.
Any other questions?
And we'll have time at the end as well, once Lori goes through some of her information. But also just, I want to, once again, reiterate, you can get a hold of, Lori, myself, through Dan, through Sarah. We're happy to have additional conversations. In the end, this is what we enjoy doing, and we want to be helpful to you guys.
Okay. I'm going to go ahead and share my screen. I'm going to go into presentation mode. Is everybody able to see my slide there?
Yes.
Okay, I'm just going to give a brief bio on myself. I've been introduced, but as you can see, I'm a nurse by training, and then switched into the clinical research industry over 20 years ago. Started as a CRA and have worked my way up through the ranks at a variety of clinical research organizations, big pharma, AstraZeneca, J&J, companies like that, as well as recently over the past, 8 years, I've been exclusively emerging biotech and exclusively immuno-oncology. So prior to coming to Cancer Insight, I was in your position. I was working as VP of Clinical Development and Reg Affairs at an emerging biotech that was trying to develop an immuno-oncology asset. So I feel your pain and frustration. So first thing I want to talk about is study startup. And I'm not familiar with how the level of experience each of you have. So my apologies if this is, a little too elementary, but I'm just going to give you some things to think about. So a study drug. So you do want to estimate the total consumption that will be required over the course of your trial, because there is nothing that can derail a clinical trial more than running out of study drug and having to pause enrollment on your trial. So it's very important if you're doing, weight-based calculation, dosing, or anything like that. You just really need to make sure that you have accurate estimates of consumption. You need to sort of have in your mind, how long are these patients going to remain on treatment? Are we in a salvage situation where it's, we're only expecting life expectancy of 3 months before they progress? Or is this, like George mentioned, a more proactive situation where these people could be followed in a setting up to, 5 years? So it's important to make sure you know that upfront, so that during the course of your trial, you're not running out of your study drug. The other thing is expiry dating. I want you guys to think about that. The general rule of thumb is
Lori?
that, yeah.
Sorry, I was just going to say your slides are in presenter mode.
Oh, you're not.
What we're seeing is your presenter mode. Yeah, Lori.
Your presenter mode, yeah.
Oh, that's terrible.
No, it's
It's not terrible, but just thought you would want to know.
No, that's not Cool. So I'm gonna try this again.
We're still seeing the presenter mode.
So display setting, do you see display setting? On your screen, Lori?
I'm looking. I'm looking.
Right there to the right.
To the right,
Like over here.
where your mouse click is. Where the cursor is, there's a display setting.
Am I not in the right place?
No, that's fine. You just need to like the display setting. Just look at your mouse cursor. Right next
Yes.
to the right, there's display setting. If you click on that,
Yeah, I'm not saying display setting. Am I blind?
Yeah, on presenter mode, I think you're looking at the Zoom controls, Lori. What he's trying to point you to is at the top of your, the black, no, not that one, sorry. At the very top of that black screen that you're looking at right now, in the middle, right next to end slideshow and show taskbar, there's display settings, and you should be able to switch which screen is displaying through that. Nope.
I know. I was just trying to get out of that to see, because I'm not seeing anything but the I'm not seeing what you're talking about. That's all.
So if you go straight up from where your mouse is right now,
Oh, I see display settings. I see it now.
there we
Yeah.
go.
swap.
Okay.
There we go. Looks perfect.
Oh, thank you for walking me through that, because sorry.
That's okay. Happens.
All right. Expiry dating. So the general rule of thumb, you would be doing ICH stability at regular and accelerated conditions. And the rule of thumb is that you can basically take your accelerated condition stability and that would allow you to sort of double your room temperature stability. So say you have 3 months accelerated stability, you could get an expiry for 6 months at room temperature. So you have to think about the expiry dating. Now, a lot of you won't have extended periods of stability when you're submitting your IND. In fact, most emerging biotechs, you'll just be submitting your IND around the time that you have your, initial and 30-day release data available. So keep in mind that it would be like a rolling sort of stability, and that you would have to keep your sites and everything updated on the expiry as you go along. You want to think about your drug depot. Where is it going to be stored after it's manufactured? Who's going to do the labeling? Are you blinding your trial? Do you need to have unblinded staff? Do you need to have a randomization scheme? All of these things are going to be important in trial design. Also, your clinical trial material packaging. There are container closure tests and things that you have to do, sterility. And so just also keep in mind the site's availability. The sites do not want to receive 80 kits of, clinical trial material. They have limited room in their freezers and their refrigerators. So making sure that your CTM packaging is also very site friendly, and that it's being given in quantities that is easily manageable by the sites. Finally, assessing the need for IRT, which is interactive response technology, or randomization and trial supply management. They mean the same thing. It's basically a randomization system that's done in case you need to add stratification factors into your trial, or if you are doing, a truly randomized trial and you want the extra control. Stratification factors might be, for instance, if you want to stratify by, PD-L1 positive and PD-L1 negative, or any other biomarker, or any other type. George, anything you want to throw in there for stratification factors?
No, I think that's good for now. I mean, we can go into that if people have questions, though.
Okay. And again, this system is a system that will coordinate between your depot, and oftentimes it is interfacing with your electronic data capture system, with your clinical database. And so remember, the build times for systems like this is generally about 12 weeks. So you're going to need to know well in advance if you need this type of build and service for your trial. The other thing that you're going to want to do is think about CRO selection and the timing of that. Now, pre-COVID, I would say that you would be fine reaching out to your CRO 6 months prior to your anticipated first site activation. At this time, post-COVID, and given the current labor shortage at our sites and their inability to quickly and efficiently activate, you're going to want to start discussions with your CRO as early as possible, no less than 9 months. And again, to George's point, including us earlier, even in the design of your trial and helping you get into the clinic and through the FDA, is going to be to your advantage as well. So I can't stress that enough. It's not too soon to start thinking about CRO selection. You also want to think about your RFP and assumptions. When you go out and are soliciting bids from multiple CROs, you want to make sure that you are comparing apples to apples. So it will be an important step for you guys to sit down and think about, what activities do I really want this CRO to bid on? And there are, real cost drivers involved with that. One of the cost drivers is something we call source document verification. Do you want the clinical research associate to verify every single field in your casebook, or do you want to have a risk-based or a targeted monitoring plan where they're only looking at critical fields? Things like that can make the difference in quotes, and you want to just make sure that your assumptions are aligned and that the CROs are competing against the same set of assumptions so that it's easy for you to decipher between them. It's always smart to do multiple bids. And then you do want to leave some time in for contracting. So just be aware of certain pitfalls. And as a former CRO employee, I can tell you that there are different levels of integrity across this business. And there are CROs that will, say, yes, 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 we can do it, and they'll give you this very, very cheap price. But then you're going to find out that they're going to come back to you and be like, oh, yeah, that wasn't included. That's out of scope. We're going to have to charge you for that. Oh, yes, we didn't include that. So we're going to charge you extra for that. So just think about that in your contracting process, and ask them specifically about their change order process and, what's involved if there's an out of scope or something that we didn't originally consider. And then last but not least, something called the TORO. This is a transfer of regulatory obligations. So this is an FDA document that you'll have to submit, generally with your IND, transferring certain regulatory obligations from yourself as a sponsor to the CRO. So just make sure that that's in place, and that way you guys are covered. So you're making the CRO take on a lot of your responsibilities as far as the regulatory requirements. Any questions before I move on? Okay. When thinking about who to select to run your study, you want to go with someone who understands emerging biotech, understands your budget constraints, understand what you've committed to your board, understanding, what you've committed to your investors, what you've committed to the street, do they feel your pain? Can they understand what you're going through? You also want to make sure that they have lots of relevant therapeutic area expertise. It's some CROs are going towards this non-therapeutic area, but I challenge them to take a team that's been running cardiovascular or anti-infective diseases and have that team run an early phase oncology study. There's going to be issues. So look for that expertise whenever possible. When I'm looking for vendors, I want those niche boutique vendors, the ones that have a super focus and that laser beam sight. They do one thing and they do it really, really, really well. You want to look at staff turnover rates. It's very high in the industry, so do not be surprised if your CROs are coming back to you with staff turnover rates in the 25% range. And with the duration of oncology studies, it's sort of inevitable that there will be turnover on your team. So what you want to do is inquire with your CRO, what is their sort of change management plan and how do they address turnover? Because it's in today's workforce and labor market, it is something that's inevitable. You also want to consider the level of executive oversight. When you have a problem, will your voice be heard? And will it be heard by the right people? This sort of lends itself to the size of the organization that you select. Obviously, the bigger organization you go to, you're going to be a small fish in a big pond. So think about when you're selecting a CRO, how accessible is the executive team? What level of oversight will they be providing? And lastly, you want to consider size, which I've already mentioned, and the structure. How dependent are they on the use of contract staff? Where are they geographically located? Their management structure, like all those things can impact the performance of your CRO. So you want to consider those when making a decision. We want to talk also about other vendors. When you think about setting up your clinical trial, and specifically when you have to come up with budgets for your boards, you're going to have to consider some of these. Central laboratory, and that could be for the analysis of blood or tissue samples. Most of the time in oncology, our safety labs, our chemistry, hematologies, and urinalysis are going to be done locally at the site themselves. And you're only going to be collecting blood for PK, biomarkers, immunoassays, and same thing with tissue. With each laboratory, you're going to have to make sure that you're providing the sites with ample kits and supplies. Those are going to, sometimes require barcoding, special serialization, special shipping instructions. So you want to consider who's going to be your lab kit vendor. We've already discussed about the drug depot and the IRT or RTSM. George mentioned before that we have a client that has some cardiotoxicity issues. Will the FDA want you to have, a central EKG reader or 24-hour Holter monitoring to evaluate for prolonged QT intervals? Will you have to do central collection of troponins? Those type of things. When thinking about your registrational trial, when you're going after an endpoint like objective response rate, the FDA almost always is going to require a central reader. So this would be a vendor that would receive all of the patient's scans, CT scans and MRIs, and they would independently conduct their own RECIST evaluation in addition to the PI, and they would be sort of like a blinded independent reader, and they would verify the results of the trial. And as you can imagine, that's a pretty heavy cost to get radiologists to go ahead and do these rereads on all of your data. You will certainly need to have IRB approval before you go in. In terms of site startup, if you can get your sites to go under a central IRB, which is a singular IRB that can provide approvals for all of the sites participating in your study, that is preferred over what is called a local IRB, where the individual institution has their own IRB and you have to go through their process. Thinking about translation services, because there are areas of this country where you might need documents translated into other languages. Spanish is a big one, so think about that. And from a system perspective, you're going to need a clinical database. There's expense associated with that. You're going to need a pharmacovigilance and safety database. There's expense with that. You're also required to have a trial master file. What system is going to be used, who's going to host it, and how much is that going to cost? And lastly, for early trial designs, dose escalations, and even for phase 3 registrational studies, you may need a data monitoring committee or external data safety monitoring board, which is generally a group of independent individuals that are contracted with to serve as, independent decision makers on, set time points throughout your trial to make safety related decisions. Talking about study startup, I want to spend a minute or two on site selection. I've mentioned local and central IRBs, and they are generally associated with community sites versus academic sites. Community sites in general have quicker startup times, but the downfall of the community sites is they may not be able to, get the quality and quantity of tissue that you require for your study. So there's a trade-off, community versus academic. Academics do have a much longer lead time for startup, so just keep that in mind and maybe go with a mix of both depending on what type of trial you're conducting. You also want to think about who are the key opinion leaders in the field that I'm going after, and what is my publication plan? Can we schedule interim analyses so that we can present preliminary data at ASCO or at SITC? And what is that going to look like? Who will be the champions of my study and represent my company? Geography. If you go to the FDA and you have all sites in California for a registrational trial, they're going to question that. You want to make sure that when looking at your sites that you have a nice geographic spread across the country for a number of reasons. Things that could impact patients, things like weather, diet, lifestyle, specialty populations. Those are all things that you want to think about when selecting your sites. And in order to select your site, first things first, get your non-disclosure agreement signed, and then you'll want to collect some formal feasibility. You're going to want to ask them what type of IRB do they have? How long will it take them to get up and running? Do they have a dedicated coordinator? How many patients do you think they can enroll per site per month? These are questions you would ask during a feasibility and get that survey done. Once the survey is in, you decide if you want to move forward with a qualification visit, in which case a CRA would go out to the site, or it can be done remotely post-COVID, where they will assess and interview and to look over the qualifications of the staff and make a recommendation if they're fit for use on the study. You'll also have to go through, at minimum, the IRB approval process. Depending on what type of agent you're using, you may have to go through an IBC if it's a biosafety sort of thing. If it's a virus or something like that, you will, in addition to an IRB, you will need an institutional biosafety committee approval. Large academic institutions, they also have a scientific review or protocol review committee that it will have to go through. And these things, they just take time. So this is why I'm saying it's never too early to engage your CRO. Something else that has been coming up is a radiology committee approval. If you're doing CT scans, if you're following RESIS 1.1, if you're doing FDG PETs, they may require that it go to a special radiology committee to determine the amount of exposure and estimate the risk benefit to the patient. And lastly, contracting and budgeting with the sites. This can be a daunting task, and there are things that are coming up that sites have figured out that they can monetize and charge you for, and prices are just going up, up, up. So your per patient budgets, they don't look like they used to, 20 or even 10 years ago. And it's not uncommon to see overhead rates up in the 30 to 50% range, especially in academia. I know for a lot of companies, first patient dosed is a key deliverable, and just touching on the gating items for that is your systems, which, again, I mentioned is going to be a 3-month lead time. Your IP availability, if your drug manufacturer is in China and you need to get that imported into the United States, you want to make sure that the IP is available and on site in order for you to get that first patient. So be aware of those timelines, be aware of how long it's going to take to get a contract and budget in place, the IRB and other committee approvals, and also sites themselves will have to complete regulatory documents, which are called site essential documents, financial disclosures, 1572s, a bunch of documentations is required prior to CROs having the official green light to actually ship drug to that. So just think about all those gating items when you're thinking about your first patient. In the maintenance period, we'll focus on patient enrollment. You need to consider whether you'll be doing any interim analysis or the setup of your safety review committee meetings, usually done, for dose escalation or, at the time of any adaptive trial changes. I will tell you that data monitoring is different today than it was before COVID-19. Before the pandemic, we used to send out all the CRAs and they would go on site and they would visit the PIs and the site staff and log into the systems and review medical records there. But since then, the majority of our oncology sites are not allowing the CRAs on site, and they're providing remote access to their EMR systems, and monitoring is being done that way. There are a couple of drawbacks to that. We're not getting to check the investigational pharmacies for, accuracy in their reporting of inventory and things like that. So there are a couple drawbacks, but on the whole, it's been a pretty seamless process and the sites have adapted to it quite well. During this time, there will be lots of data cleaning activities during study maintenance. You're going to, want to make sure that your CRO is providing you with monthly updates on how much monitoring is being performed and that data is being cleaned on a regular basis. What you don't want is to come to the end of your study, have all your patients enrolled, and then figure out that you've got all these discrepancies and you've got to start issuing, a bolus of hundreds of queries when you're trying to lock your database. You also need to reconcile your database against any third-party systems. I did mention a safety database, so all of your SAEs are typically housed in a system other than your EDC by a pharmacovigilance vendor. And so making sure that the terminology and the grading is consistent across is important, as well as any central labs. You're going to want to make sure that things like, patient identifiers and visit dates are consistent between multiple system and data sets. You'll continually have to monitor IP supply because, as I mentioned early on, there's nothing that derails a trial like running out of study drug. And we recently had a case study example where we enrolled way quicker than ever anticipated. and had to pause enrollment to allow time for additional study drug to be manufactured and released. And that's never a good thing. Lab sample management. You do want to make sure that your biomarker strategy is clearly defined up front and know what you want. And if there are critical pieces like tissue samples, you want to make sure that that's being adequately tracked and followed, and that results are being made available in a timely manner. One of your deliverables at the end of your study is going to be the trial master file, as I mentioned. And it's going to be, again, one of those things like the clinical database. You don't want to be in a position where at the very end of the trial, you're trying to go back and reconcile and find missing documents and request missing documents. So maintaining that is essential throughout the life of your study. And there's also regulatory reporting requirements. You will at minimum have to be submitting to your IND on an annual basis for safety, but you'll also have to report any CMC changes, and as part of safety reporting, any SUSARs or other required reporting events. And then last but not least, study closure and reporting. As a CRO, it's our job to deliver to you a final lock database, after which you're going to have to close the site and close down the IRB. You will have to pay a continuing review with your IRB every year. So you don't want to get stuck with an extra bill if you forget to close out with the IRB. The study trial master file is a deliverable from your CRO, so you'll be required to maintain that. After the database is locked, biostats will begin their analysis and provide ADAM and SCTM data sets to complete your full tables, listings, and figures that will be required for your clinical study report. You'll most likely want to publish your results in a manuscript. And last but not least, there is a requirement for most studies. There are a few exceptions, but for most studies, you will have to report your trial results on clinicaltrials.gov. So any questions?
So let me just make one kind of concluding statement, and I'm happy to answer any questions or discussions. I hope we haven't depressed you guys with all of this information, because obviously there's a ton of requirements when it comes into clinical trials. Any human subject research is highly regulated, as all of you are aware. And because of those complexities, you really have to think of your CROs, your clinical research organizations, as the group that knows the rules, know how to do this kind of work. And I don't like to put a negative connotation on it, but it's an essential element. I'm going to avoid the necessary evil part, but it's an essential element to getting your studies done, quite frankly, because you can imagine if you tried to recreate everything that we do every day, it would take you a lot of time, a lot of effort, a lot of money. And the reality is, you just need to find a good partner that can help you through the process, know the rules, know how to conduct these early oncology studies. And that way you can feel confident that they can guide you through the process. There's no reason why you have to understand absolutely every one of these elements that Lori's described. That's what CROs do. So anyway, I hope that this overall has been helpful as an overview. One, how to start thinking about your trial, never too soon to do that, some of the elements that go into it, but do understand it's a very complicated, very detailed area that requires some kind of professional guidance through the process. And of course, there's heroes out there that can help you do that. So questions, any discussion points, any clarifications we need to make? I know we're kind of at the end of our time block.
Yeah, I have a question here. So
Yeah,
thank you very much for the presentation. It's very informative. Indeed, it's a very complex process. So my question is that I think for many of us, So probably we are not at the clinical trial stage, but at the planning stage, right? So we want to get a grant either from CPRIT, from AIH, from other, even private foundations. So I guess my question is that with you, how we can proceed? Say that, okay, can we reach out to you and say we sit down together and discuss some details, let you understand the project, and then we create a plan together and to get the fund, and then we can proceed to do the work.
Yeah, and Dan has probably told the group before, if he hasn't, we've worked with a lot of companies on CPRIT type applications just to address the things that you're describing. Because at this point in time, you're just starting the process. You have a compound, you're not quite sure what your clinical development plan is going to be. However, for those non-dilutive fund applications, they want to know that you have a plan, right, and you've given it some thought, and you at least have enough details in there that they can pressure test it and say, okay, yes, these guys have an understanding of what's in front of them. And they've given it adequate consideration, and therefore it's something that is fundable. And that, and I'm not sure how far you guys have gotten into this, Sarah, with the CPRIT applications, but you do not have to have a full-blown clinical protocol, for a CPRIT application. A lot of the companies in your stage, what they're looking to see is, do you have a plan for your preclinical plan that informs your clinical plan? And your clinical plan is sufficiently outlined that you understand what all goes into it, So it doesn't have to be the full cooked protocol. Typically, a trial concept or even a synopsis is more than sufficient for a CPRIT application. So, and we've probably written, I don't know, hundreds of these now. It's something that we've done repetitively. So, short answer to your question is, we're here to help. We're happy to engage. We can have those conversations. We can help you with your applications.
Okay, that's great. Thank you.
And George, to your point, yeah, not meant to scare anyone, but too many times I've seen CEOs sort of who don't understand, and they come in with a little bit of naivete, and they commit to investors or to their board certain milestones, like, yes, we'll have first patient dose. And that's one of the things that we struggle with the most with our sponsors, is there's just a little lack of understanding of how complex clinical development is. And so just sort of giving you a, here's what you ought to think about. And it's clinical research is not for the faint of heart. So just wanted to, our job is to help prepare you and prevent you and make sure you guys are each successful. And I don't ever want any one of you to have to go to explain to a board member or investor why you didn't make a milestone. So that's kind of what we're here to help with, you guys to be successful.
Hey, Lori, I wanted to jump in and ask you a question, just sort of building off that conversation. So in your all's mind, how early, is there such a thing as too early to start talking to groups like yours? And what kinds of, like, let's say they do sort of set up an introductory meeting with you and your team, which I would highly recommend to everybody here. What kinds of information is useful to bring to make that a very productive conversation versus what do you see people kind of getting stuck on sometimes?
Lori, before you answer that question, or as you're formulating your answer, I do want to, one quick caveat. If you go and just ask your typical CRO, hey, will you help me? That's not what we do. What we do is we run clinical trials. Give me your trial and we'll run it for you. So there are more specialized groups that do the early development and are willing to engage with you for these early conversations. So I just want to make it clear that your typical CRO may not be able to engage with you this early on. However, it is important to find people who can engage with you so that you can start thinking about this clinical plan. And Lori, you want to add to that?
I think you've touched on it. We've shown you how complex and involved this is. And, you want to get the input of subject matter experts the sooner the better.
And then, Sarah, the second part of your question, as far as the materials, like if you came to us and said, you know, we need you to help us figure out a clinical development plan and a strategy. The things we need to know from you are your drug, your technology, what its mechanism is, where you see it having its best benefit. Now, you may tell us this is going to work in or we'll just keep with our example. This is going to work in triple negative breast cancer, right? Great, but what do you need from these patients? Are they accessible? Or, you know, is this going to be feasible? We'll go through that with you. And you may say, well, it will also work in ovarian cancer. Well, guess what? There's going to be more of those available for your particular drug, you know, given the requirements. So that's where we start guiding you towards that target patient population. But anyway, your technology, what's the mechanism? What do you see as the patient, that population that's most likely to work on? Any preclinical data you've already generated to support that. Where you are exactly in your development path. Some of you may have not even started yet. You may have, you know, just have a concept. Others of you may be ready to file your IND, you know, so there's a lot of ground to that, you know, that can be covered between concept and IND submission. So where are you? What information you have? That will be helpful in that discussion.
All right, thank you. Are there any other questions from the group here? Because we're right at
Yeah.
time.
Yeah, I have just
Yeah.
one question. This is related to separate application. Just, you know, for the purpose of budget preparation, I wonder, can you give us a ballpark number? How much it may cost to get you involved in you know, guiding us through the phase 1 study? Some idea?
Just for the consulting part of it, or, you know, delivering a protocol, or are you talking about the actual clinical trial cost?
Maybe both
Yeah.
towards clinical IRB protocol development. And then
So
first
the
phase.
way we typically structure things, if there's things that you need assistance with, we can do something as simple as a simple consulting arrangement where it's just an hourly rate to have some conversations around design and concept and such. And so it's a relatively low cost, you know, venture for you guys just to get the discussion going. If there is more, like we want to review, you know, materials and start thinking about what it is, you know, we're doing for our IND and or perhaps a pre-IND, and, you know, do we need to fill in gaps? And now it's getting a little more involved. Then we do have regulatory guidance to provide there, and we assemble a small team that will work with you to look through your pre-IND, IND strategies. And quite frankly, we've filed several dozen of these now. So, you know, we're happy to lead that process. Once you get to the trial phase, the way that typically goes as you're interviewing your CROs is you have your full protocol, and the protocol and the CROs will literally bid on that and tell you, it's like, you know, building a house. Here's the plans, how much is it going to cost me? And they will give you a number of what that clinical trial cost will be. To Lori's point, clinical trials themselves have just, like practically everything else that we pay for these days, just the prices just continue to rise. And so, you know, there was a time there where we told everybody, you know, plan on, you know, $50,000 a patient to do a phase 1. Well, that's almost doubled. So just to kind of give you a sense, we're in the 85 to 100,000 a patient to do a full-blown phase 1 with all the correlatives. So, you know, once again, you can do stripped down versions of it, but it's getting very costly. And I think everybody just needs to be aware of, you know, where this is going. It used to be you could do trials for under $1 million. It's hard to do that these days. So
Thank you.
once again, I don't mean to depress you guys. I'm just trying to give you a sense of reality out there. Okay, so it used to be you could develop drugs in your basement. Well, we're past that point now.
So also would like to get your take on, you know, lots of academic institutions like the Anderson Cancer Center, they do have phase 1 program. It's usually physician sponsored. So how do you weigh that option versus a CRO?
Yeah, no, that's a great question. And let's just use MD Anderson as an example. They have a full-blown phase 1 program. Now, depending, they will do a lot of the CRO type activities within that phase 1 program, but typically for internal trials, not external trials.
Single center.
Yeah. Now, there are also phase 1 trial centers. Let's use, Next Oncology in San Antonio. Phase 1 trial center, that's what they do. And they can provide you most of the services around that phase 1 trial. However, MD Anderson for external trials, no. Next, depending on, is it a single institution versus multiple, a few other variables there. A lot of times what will end up happening is you have to engage with a CRO to manage all of the activities, and then MD Anderson or Next are acting as the site, and they're the ones that are actually enrolling and treating and following those patients, but you still have to have a CRO covering all this, myriad of activities that go into, in the end, giving you a validated data set and TMF that you can use to file with the FDA. So.
Thank you.
All right. Why don't we, that sounds like this seems like a really good sort of finishing point for today. As you guys know, there's going to be a lot of conversations as you really start thinking about this and think about how it applies to your own conversation. So just to say it one more time, always recommend that if you want to set up a one-on-one, take advantage of that, at least to sort of get better introduced and talk more specifically about what you're working on. With that, thank you so much, George and Lori. You both really just shared a lot of knowledge again today, as per usual. So always appreciate having you join us.
Thank you,
Okay.
Sarah. Thank you.
Thank
Thanks, guys.
you.
Thank you.
Appreciate it.
Thank you. Thank you.
Thank you.